Good evening, my fellow scientists. It is Thursday, November 16th, 2017, and I want to talk a little more about CRISPR-Cas9. I said yesterday that I was not aware of any trials in which CRISPR-Cas9 had been added to humans, and it turns out that was half true. There has been one approved clinical trial in China where CRISPR-Cas9 was used to alter the genes of human cells, which were then reintroduced to a human. So it's a little bit different than the animal study I referenced earlier, where CRISPR-Cas9 viruses were introduced into an organism, an adult organism, which was then modified in situ. I mean, you can imagine that if someone had liver disease, it would be great if their liver could be altered by some external medicine, essentially, that could change that genome. That would be a lot better than taking a few liver cells, modifying them, and attempting to grow a new liver for that person. But that strategy can work for certain blood diseases, and it could conceivably work for some cancers, where in the case of the Chinese clinical study, cells were pulled out, modified, reintroduced. They're blood cells, so that's not a huge technical problem to take them out, grow them up, and reintroduce them. And they were modified in such a way that they recognized cancer that they had otherwise ignored, thus hopefully curing that cancer. As of yet, there are no results of that trial posted publicly. It's still ongoing, but it's been going on for a year. So the fact that it's not closed yet may or may not be encouraging. So CRISPR-Cas9 has been used to edit human genes one time. There's been some reports that maybe it has been used to modify human embryos, though that those have not been grown in any substantive way, but the technology is definitely capable of modifying human genes. So having looked into all that, I think that my assessment that human trials of CRISPR-Cas9 in situ, those have not happened yet, but ex-vivo modification with CRISPR-Cas9 had. I was not aware about that, but now I am. So clearly the technology is moving forward and will be even better every day. Where I find that exciting is where genomic studies like that being organized by George Church, looking at Supercentenarians, people who've lived more than 110 years, looking at their genes in total, and then saying, well, if we have genetic changes that are correlated with longevity, could we introduce those kinds of changes into people directly, into living, breathing adult people? And to do that, you'd have to introduce the genes in some sort of vector. And as of yet, that has been a risky endeavor. This new zinc finger endonuclease virus that was undergoing clinical trials now in humans means that it's being resolved, that there may be solutions, but I found the reference to the 1999 study that resulted in the death of the patient, so you definitely can have runaway immune responses to these kinds of things that can be fatal. So there's risks and benefits. It's beautiful to think we could live to 110, not so great to think we could have systemic immune shock and just die. So anyway, links to all that are in the description. Check those out if you're interested. It's a beautiful new field. Stay tuned Monday through Friday. We talk about chemistry and gene editing and science news every day right here in the Allen Lab.